So I'm James Davies. I'm a consultant haematologist and scientist at Oxford University, and I'm just going to do a brief presentation on the new ways of treating sickle cell disease and thalassaemia with a new technology called genome editing. So the first thing really to appreciate is how big the genome is and why this is such a difficult task. And this is a printed copy of the genome. There are actually 3 billion characters in the genome, and if you print it out, it covers a huge number of volumes, as you can see, and each of the different chromosomes would need to be printed out in several very large books. And actually, it works out to be about the same size as the entire of Wikipedia in terms of the amount of text that's in it. And the difficulty with fixing sickle cell disease is that you've just got to change one of the letters from a T to an A. And then you need to convert that single letter back again without changing any of the other characters. And you need to do that in several million cells at the same time in the bone marrow-derived stem cells in order to fix the disease. And that's actually proved extremely difficult to do until very recently. So, as many of you will know, you can actually correct sickle cell disease by collecting stem cells from somebody without sickle, and then you give the patient chemotherapy, and you can cure people of sickle cell disease with this technology. And we've been able to do that actually for the last 20 years or so. The problem is that you need to have a sibling really who is a match, and there's only a 1 in 4 chance that a brother or sister will be a match. And unfortunately, the safety of transplantation reduces very substantially as you get older. So it's only reasonably safe to do it in children. And unfortunately, large doses of chemotherapy are required, and there's a very real risk of the donor cells attacking the patient, and that can lead to graft versus host disease. And so really it's not a reasonable treatment to undertake a bone marrow transplant unless the patient's got very severe sickle, which is picked up in childhood. Now, there are 2 different types of transplant that we can do. And there's a type of transplant called an autologous transplant, in which the stem cells are collected from the patient and then given back to that same patient. And that's a much safer procedure than when we do it with stem cells from one person going into another. And we've always very much hoped that we'd be able to do this for sickle. And the idea here would be we would collect the bone marrow stem cells, which give rise to all of the different components in blood, change the genetic code very specifically at the one site that causes sickle cell disease, and then put them back into the patient to cure them. And this has been done very recently. So the first patient was treated in November 2019. She is a lady called Victoria Gray, in my opinion, an exceptionally brave woman, who was the first person in the world to have a bone marrow out transplant with genome edited cells. And it's the first time that this technology has been successfully used in humans. And this was done in Nashville, Tennessee. And so how did they do it? Well, with the first generation of technology, it's not actually possible to correct the sickle mutation itself, but we know that sickle is caused by a mutation in the beta globin gene, which is the adult form of hemoglobin. But in virtually everybody who's got sickle, there's a perfectly good globin gene called the fetal hemoglobin gene that's just next door to it. And unfortunately, this gene gets switched off by a protein called BCL11A within the first 6 months of life. And then the person then runs on the adult version of the protein. And for many years, people have wanted to upregulate the fetal hemoglobin in patients with sickle cell disease. And this is what this technology does. And what it does, and the way it works, is that the edit is actually made in this gene BCL11A, which switches off fetal hemoglobin. And the idea is that you make a deletion in the switch with this thing called CRISPR-Cas9, which deletes the switch that turns this gene on, and therefore that gene doesn't bind to the gamma globin promoter any longer, and it upregulates the expression of the fetal hemoglobin. So much more fetal hemoglobin is made than normal. And what are the results of the initial studies? Well, this lady had 7 sickle cell crises per year before the study. She's now free of sickle cell crises. Her blood tests are similar to someone who's got sickle cell trait. She's got hemoglobin levels of 113 grams per litre. 95% of her red cells have been edited, which is incredibly exciting. And she's got nearly 50% hemoglobin produced from the fetal gene, which would effectively make her have a sickle trait. In terms of the complications of the transplant, she had infections whilst her immune system was down, and she developed gallstones. It took 30 days for the new bone marrow to make blood cells again, which is actually quite a long time. And that's longer than we would ideally like for safety reasons. And the full safety of these types of genetic therapies won't be known for many years. The other thing to say is that this is only the first generation of the technology. So we've been working on directly fixing the sickle mutation itself. And this can be done with a second generation of genome editing technology called base editing. And what you can do here is this is the normal, is to have an A here. In sickle, it's a T. And what we do is convert it to a C. It's difficult to convert it for sort of complicated reasons back to the normal sequence, but we know that if you convert it to a C, it makes a form of hemoglobin called hemoglobin G mercapsa, which there are a few people in the world have, and they are absolutely fine. So people with hemoglobin G mercapsa have got normal hemoglobin, their cells are normal size, and they have a normal amount of hemoglobin in each cell. And these patients have no evidence of any sickling. And there are people alive with just a single mercapsa who've got 100% hemoglobin mercapsa, and they're also fine. And we've been able to replicate this in the lab. So you can see here, this is the sickle mutation, which is a T, and we've converted nearly half of those cells to Cs. And this technology is improving day by day, and I think very soon we'll be able to nearly get 100% editing at this site. This is all very exciting, but this is very early days for this technology. And I guess I like to think of it a little bit like mobile phones. This is sort of massive handheld mobile phone that we had in the mid-1980s. And we've got a bit of a way to go till we've got beautiful smartphones. But I'm very hopeful that this technology will allow us to cure a large number of patients with sickle and thalassaemia actually in the near future. And that leaves me to really to thank all of the people who have been involved in the research, particularly the patients that have given us blood and bone marrow, and also to thank all of the researchers, particularly my PhD student Mohsin Badat and Peng Hua, my postdoc, but also the very senior people who've helped fund this, particularly Noemi for supporting the research, and Doug Higgs.